So our third speaker is Dr. Jingyuan Chen. Dr. Chen is an assistant professor of radiology at Massachusetts General Hospital and Harvard Medical School. Her talk will be Simultaneous EEG-PET-MRI identifies temporally coupled, spatially structured hemodynamics and metabolic dynamics across wakefulness and REM sleep. Due to the ongoing situations in the US, so she also cannot make it, so we will play a video recording of the talk. Thank you very much.
Thanks the organizer for inviting me to join this symposium on the relationship between brain functional organization and glucose consumption. Unfortunately, I'm unable to attend this year's conference in person, so I have recorded a presentation instead. I have nothing to disclose. I'm sure you have gained valuable insights from the previous two talks exploring the correspondence between the brain's metabolic landscape and the functional patterns during the awake resting state. And now, since it's a morning symposium, and given jet lag or lack of caffeine, it's quite possible that your brain is in a drowsy or sleepy mode. So you may find the talk interesting because we will share our latest work on how glucose metabolism and brain organization change during sleep. So it's well established in the animal literature that sleep is accompanied by substantial changes in various metabolic processes, including glucose consumption. And in humans, brain dynamics during sleep have also been extensively studied using simultaneous EEG-PET-MRI. A common observation is that during non-REM sleep, particularly in the light and intermediate stage of sleep, the amplitude of fMRI-based hemodynamic oscillations increased dramatically. And these large fluctuations also become more spatially coherent, so we may see an apparent increase in the functional connectivity metrics. So despite a large number of work mapping sleep-related metabolic and hemodynamic changes, these physiological processes are often assessed in isolation. So this poses some challenges for evaluating their dynamic coupling in real time. So the goal of this study is to characterize the spatial temporal dependence of functional and metabolic dynamics across different arousal states. So temporally, our neuronal hemodynamic and metabolic processes were coordinated during the descent into non-REM sleep. And spatially, what brain-wide metabolic changes support the altered patterns of functional activity during sleep? So to address these questions, we basically image everything together. So with EEG, we can now have grand truth delineations of each subject's arousal state. And with fMRI, we map whole brain hemodynamic oscillations, which indirectly reflect functional activity. And finally, to capture dynamic changes in glucose metabolism, we use a novel functional PET-FDG method, FPET-FDG. So briefly, FPET-FDG is a technique introduced by Jacob Hooker's group at MGH about a decade ago. And we know that in conventional PETs, we use a bolus injection of FDG tracer. And to quantify stimulus-driven changes in the metabolic rate of glucose, we really perform two separate scans, one doing task and one for the baseline, and perform the comparison. But FPET-FDG replaces the bolus injection with a constant fusion paradigm, such that the PET time activity curve will gradually reach a steady state, where the slope of the curve is proportional to the steady state metabolic rate of glucose. And now we have a task. In this case, the time activity curve will reach a new steady state, with a new slope corresponding to the new metabolic rate of glucose. So it manifests as an altered slope in the signal. So in this manner, functional changes in metabolism can be captured within a single imaging session. And we can also use general linear model to infer actuation in FPET data. So just a quick comparison, unlike in fMRI, where the baseline is relatively constant and the signal returns to baseline after a change, the baseline of FPET is roughly linear, and the task-induced changes will reach a new intensity level following the stimulus, because FDG will just be trapped in the tissue. So we also need to know the timescale differences. In this study, we primarily focus on minuscale metabolic changes here. So here's a demonstration of applying FPET-FDG to map visual activation. So compared to prefrontal reference regions, the time activity curve in the visual cortex demonstrates an activation. So we can identify a change in the slope when the flickering checkerboard is on. And such difference is more salient if we remove the linear trend from the FPET time activity curve. And the validity of this technique has been demonstrated in several studies involving various sensory and cognitive tasks. And you will hear more about its applications in characterizing metabolic connectivity in the next talk by Hamish. So for the rest of the presentation, we will only focus on the slope change, basically show the time activity curve post-detrending. So having introduced the imaging techniques, we conduct the experiments on a 3T Siemens scanner with a burn pad insert at the Martinez Center at MGH. And all the scans took place in the afternoon, and the subjects were partially sleep deprived to increase the sleep pressure. And to track dynamic changes in glucose metabolism, we use the FPET technique, and then we reconstruct the PET data at a 30-second temporal resolution. And we use a very conventional fMRI protocols. So here shows a summary of sleep state distributions across subjects, and most participants underwent stable wakefulness and sleep inside the scanner. And as you can see, subjects tend to be more wakeful at the beginning of the experiment, and as time went by, there was an increased likelihood of falling asleep. So first, let's show that our trimodal measures have sufficient sensitivity to detect arousal-induced changes in different physiological processes. So here is a spectrogram of an occipital EEG electrode, and then we use the power of alpha wave activity to infer subject's arousal state. As you can see, the subject shifted between wakefulness and sleep during the session. So consistent with previous reports, we show a marked increase of fMRI signal in the sensory cortex during sleep. And for the FPET dynamics, compared to the initial wakeful period, highlighted by the shades here, there is a decreased metabolism during sleep, evidenced by the reduction in the slope. And it had a minor increase during the brief wakeful period, highlighted by the green arrow, and reduced when the subject fell asleep again. And in another subject, where we can even identify altered hemodynamics and metabolism across different stages of sleep. So these examples suggest that we can reliably detect sleep-induced changes in EEG, hemodynamic, and metabolic signals. Then we ask the first question, whether these various physiological processes show coupled evolution over time. And one challenge we have here is hemodynamic oscillations occur on the time scales of seconds, whereas glucose metabolism captured by FPET-FDG unfold over minutes. And to integrate information across these distinct time scales, we perform the following analysis. So because the amplitude of fMRI oscillations is a sensitive measure of arousal, so we use that as a matrix of interest here. And we tracked dynamic changes in the amplitude fluctuations of fMRI signals over time by quantifying the time-windowed standard deviations. And the window length was chosen to match the temporal resolution of reconstructed PET signal. And here is an example from a single subject. The top panel shows how the amplitude of fMRI oscillations varies over time. And to mimic the accumulative nature of FDG and how it's trapped in tissue, we integrate these fMRI activity measure over time to approximate the FPET signals. And then we apply temporal detrending to remove baseline fluctuations, yielding an fMRI modeled FPET dynamic signal. And I was quite surprised to find that these fMRI modeled measures closely predict the actual FPET dynamics and even can capture very subtle changes in its variation, not to mention the major sleep-wake transitions, highlighted by the red arrow here. So not surprisingly, at the group level, a strong correlation between FPET and fMRI signal was observed, with large hemodynamic oscillations emerging while metabolism declines. So we see a negative correlation here. And the temporal coupling between hemodynamic and the metabolic fluctuations might be attributed to their coupling with continuous changes in arousal. Therefore, we also quantified continuous arousal metrics from EEG by taking the ratio of alpha wave activity to lower frequency delta and theta power following a previous study. And we indeed observed a significant correlation between these continuous gradual arousal metric with both fMRI and the metabolic activity. So these findings together suggest that the multiple physiological processes are tightly coupled and well coordinated during the transition from wakefulness into non-REM sleep. So how about the spatial dependence? We next examined the spatial distributions of altered hemodynamic and metabolic dynamics during sleep. So to assess changes in fMRI oscillations, we estimated the amplitude of fMRI signal and compared fractional changes between wakefulness and sleep. And the most prominent increases in fMRI signal amplitude were identified in sensory regions, including the visual, auditory, and the motor cortices. And for metabolic changes, we quantified the slope of fPET signal changes during sleep. And in a steady state, the slope of fPET signal is roughly proportional to the cerebral metabolic rate of glucose. So consistent with their large oscillatory activity, and the sensory networks showed only modest reductions in glucose metabolism. And by contrast, higher order cognitive regions, particularly the default mode network, showed the most substantial metabolic declines. So given the timescales of our measures, fMRI primarily capture fast oscillatory activity, whereas fPET-FDG in our application more robustly track baseline activity over minute-long scale. So taken together, these measures kind of reveal a picture of two distinct patterns of network activity. While most networks show reduced metabolism alongside increased fMRI oscillations, the sensory region show more prominent alternations between high and low activity during sleep. And these large fluctuations also results in a relatively elevated baseline that captured by fPET metabolic measures. So what might be the potential implications of these observations? And we noticed that the emergence of large oscillatory activity alongside reduced baseline metabolism aligns with the LC activity pattern reported during sleep in mice. And this burst-like LC activity have been considered to reflect periodical shifts between low and high arousability. And a roughly 50-second cycle of alternations between heightened environmental awareness and offline internal processing has been reported in several animal literature. And supporting the relevance of this to our data, we found that the prominent fMRI oscillations in our data also peaked at similar frequencies. And consistent with animal literature, we replicated findings that the global fMRI signal is strongly coupled to EEG sigma band activity and the cardiac oscillations linked to cyclic arousal patterns. As for the spatial patterns, we also examined fMRI activity time locked to EEG features associated with sensory gating, like spindle, or autonomic arousal, like K-complex. And we also found that the visual and the motor indeed show the most pronounced changes. And when we look into literature, these patterns also to some extent have already been identified. So taken together, this network-specific activity may reflect a mechanism for sensory alerting during light and intermediate sleep. So where periodic bursts of activity can enhance the sensitivity to the external environment, allowing the brain to monitor for potential sensory cues that could trigger awakening. So to summarize, we first show that large hemodynamic oscillations emerge as metabolic activity declines during the descent into non-REM sleep, both of which tightly tracking EEG arousal dynamics. And especially, we identified network-specific functional and metabolic patterns during non-REM sleep, particularly in light and intermediate stages, where sensory networks show preserved metabolism and periodic fluctuations between high and low activity, while higher-order cognitive regions are suppressed. So this organization supports a potential pathway for sensory awakening during non-REM sleep. And finally, from a methodological standpoint, our data also demonstrate the potential of trimodal EEG-PET-MRI imaging in elucidating the dynamic neurohemometabolic states underlying cognition and consciousness. So while here we focus on sleep, you might also be curious about how the relationship between glucose consumption and brain functional organization change across different tasks and disease. So my student Shirley has a poster on this. If you're interested, you should stop by on Friday and Saturday. Okay, thank you. If you have any questions about the talk, feel free to reach out by email.